Hi class, and welcome to this week's topic of meiosis. Meiosis is used to create sex cells, which are known as gametes, so let's dive a little bit into that. Okay, so our overview. Meiosis deals with the production of sex cells only. We call those gametes. So your sperm or your egg are going to be a gamete. The basic plan is to go from one cell dividing into two cells, dividing again, and then becoming 4 cells with half the number of chromosomes as the original. So we're basically saying we're taking one cell from your body and making a cell that has half as much. Because when you combine the sperm and egg, you want to have the same amount of chromosomes that you had to start out with. So there's a little bit of terminology before we get totally started. You heard me talk about haploid. Haploid is a cell that has half the number of its original chromosomes. So in a human, that would be 23 chromosomes. Now, diploid are cells with the normal number of chromosomes. 2 sets inherited, one from your mom and one from your dad. So we get half our cells from the egg and half from the sperm, which fuse together to make each and every one of us. A couple more terms. We have synapsis, which is a pairing of homologous chromosomes. You're going to have this happen throughout meiosis. And we refer to tetrads. Those are just your homologous chromosomes, so I don't want you to get confused. And what is our homologous chromosomes? That's 1 chromosome from your mom, 1 chromosome from your dad, and we generally see those pair up. So you're going to hear me talk about independent assortment. It's the random separation of those chromosomes. When we go through the steps, just like we went through in mitosis, you will see the chromosomes split. And when they split, that is them becoming separated into different cells. So another one you might want to know, spermatogenesis is creation of your sperm. And oogenesis is creation of your ovum. Okay, so I want you to copy down this diagram. This is our life cycle. So as you can see, right around here, we have a zygote. When that zygote has been fused from your sperm and your egg are haploid. And that zygote is becoming diploid. So we are fusing the chromosomes from your sperm, we are fusing the chromosomes from your egg. We then grow into a juvenile, a smaller portion or a smaller being of adults. And we still have 2N in this phase. We're going to go through mitosis in order to gain the amount of mass that an adult has. Once you are an adult as a male, you're going through spermatogenesis. After puberty, a male will go through spermatogenesis, which creates his sperm. Now in women, it's totally different. Women, actually, it occurs before you're even born. All of your eggs are made before you're born. So women are a little different than men are in this respect. Now, let's take a look at our eggs. So we have interphase, which we had in mitosis, but an interphase here is going to be the same. But we're going to go through meiosis 1 and meiosis 2. And in meiosis 1, we're going to have 2 daughter cells or 2 daughter nuclei. And then in meiosis 2, we're going to wind up with 4 cells. As you can see, these cells, or these chromosomes inside these cells are changing quite dramatically throughout the course of meiosis. And we're going to go through exactly what's happening. So our first step after interphase, remember, is just going to duplicate your DNA, and it's going to duplicate your organ. Okay, so we start with prophase 1, and prophase 1 is part of meiosis 1. This is where we're going to replicate homologs, or our homologous chromosomes, and they're going to find each other, pair up close together, and they're going to form the tetrads. So each and every one of these X's in here is a tetrad. So this is where crossing over is going to occur, and crossing over helps with our genetic variability. So let's go into a little bit of what crossing over is. So this is when portions of each chromatid or homologous chromosome break off and join adjacent chromatids on those homologous chromosomes. So This is going to cause genetic variation. This is one of the reasons that you might not look exactly like your brother or sister. It's due to this crossing over of chromosomes and genetic variability. So as you can see, we have the red chromosome, we have the orange chromosome, we have the yellow, and we have the green. And what's going to happen is they're just going to become very close. They're going to intertwine, and then pieces of them are going to break off and join to adjacent chromosomes. And as you can see over here at the end, those adjacent chromosomes are having pieces of another original chromosome on them. Okay, metaphase 1. Metaphase 1 is going to seem a lot like mitosis. We're just going to move to the metaphase plate, and we're going to have the spindle fibers attached. So it's very, very similar to mitosis in this respect. We have anaphase 1. The spindle fibers are going to pull apart the tetrads and homologs, and they're going to move to the opposite ends of the cell. So here's where we start getting into independent assortment. We're going to go through telophase 2, and then I'll talk a little bit about independent assortment. So the chromosomes start to unwind, and a nuclear envelope forms for a short time, and the spindles disappear for a short time. So while that is happening, we have duplicated these chromosomes in interphase, and we have no way of knowing which chromosome is going to go to which cell. So this is part of our law of independent assortment. Okay, so now that we have assorted two pairs of our chromosomes into those different cells, we're going to go through prophase again. And we're not going through interphase this time. If we were going to go through interphase, that would make it mitosis, not meiosis. And remember, in meiosis, we are creating sex cells. We want half the amount of chromosomes that we have in our normal cells. So, prophase 2, the chromosomes become visible again, and the nuclear envelope breaks down, and we are ready to divide again. So from those amount at the end of telophase 1, we're taking those chromosomes and we're going into prophase 2 without duplicating anything. Okay, so we're going to go to metaphase 2, and metaphase is just like in metaphase 1, we're going to line up in the middle. Nothing special about this phase, very similar to what you have learned before. And anaphase, we're going to pull these apart, and we're going to start getting back into our law of independent assortment. So in telophase, we are going to form new cells through cytokinesis, and those chromosomes are going to unwind. Now, we only have half as many because they've been pulled apart in anaphase, and then our cells are going to form around those. So it's very important to know that we have gone from 2N, or diploid, to N, which is haploid, and in a human, that's only 23 chromosomes. So the new cells form with half the amount of DNA of the original cell. We now have 4 new cells that are haploid. Now, those 4 new cells happen for spermatogenesis. In oogenesis, it's a little different, but I want you to know that in oogenesis, we get 1 ovum for every meiosis, and we get 3 polar bodies. Those polar bodies are not usable. So it's very important to know spermatogenesis makes 4, and oogenesis only makes 1. Okay, so let's talk a little bit about what we just went over. Meiosis produces gametes. Gametes are your egg and your sperm. It starts with a diploid, so one of your regular cells has the full amount of DNA, and it ends with a haploid cell. Now, let's think about when we're creating a new organism. We only want what's in a normal cell when we form that new organism. So we need half the amount if we're going to join an egg and a sperm in order to make this new organism. So haploid is half the amount of chromosomes, whereas diploid is the full amount. So haploid is 23 chromosomes in a human, diploid is 46. Each gamete is different due to independent assortment. So we have no control over where that gamete or where those chromosomes are going in order to make that gamete. It just happens and it is assorted. No one knows why. And then we also have crossing over that occurs in that phase, prophase 1. And that crossing over is going to give us genetic variability as well. Now, what happens next? We've created our egg and our sperm. So we want to create a new organism. That new organism, when you add n plus n, you get 2n. Now, only a sperm can fertilize an egg. So you need a sperm and an egg to create a zygote, which is the new cell. That's the 2n cell. An egg that is fertilized by a sperm is diploid. So you have all of those chromosomes. You have half of them from your mom, from the egg, and half of them from your dad, the sperm. Okay. So once fertilization has occurred, we have made a new organism that will grow into what we know as a baby. And then that baby's going to go through mitosis in order to grow into a full adult. So meiosis is used to create our sex cells, which we use to create new organisms. And mitosis is used to duplicate those cells. Okay, class, I hope you have learned a lot about meiosis, and we will be going over this in class next week. So I hope you have copied down all these wonderful diagrams and bring any questions related to this topic to class. Thanks so much. Bye.